Hello, my name is Mr. Chipman. Today we're going to be looking at Unit 0, Science Practices. This is AP Biology. My name is Mr. Chipman. I already said that. I teach biology, AP Biology at Murray High School in Murray, Kentucky. And this is Unit 0, Science Practices. It's kind of like a collection of the science practices that we need to know in AP Biology. It's not a unit that you'll find in the course description, but one I've kind of cobbled together on my own that kind of collects the different things. So, we're going to look at it. Let's do that. If I can get it to work. Here we go. All right. So, first thing we're going to look at is this idea of data representation. A whole lot of this class is understanding and being able to look at a graph or a table like you see here and being able to make determinations on those things. I have no idea what marks are at all or what these marks are, but I can see that there are 5 students that are being measured and that their marks are over here on the y-axis, and you can compare them because this is a bar graph. I literally have no idea what this table is, but it's a collection of raw data that you could then organize in a different way. And so a whole lot of this class is going to be looking at data sets and then being able to make determinations on them, being able to draw information from them. And there are different kinds of graphs that you're going to use to do that. Here they are. So I'm going to zoom in on these to help us a little bit. Here we have a bar graph. A bar graph is a graph that you would use to compare categories. So in this particular graph, we're comparing types of fruits, right? The number of people that, I guess, chose which one is the nicest. I don't know what the nicest fruit means, but it's a comparison, right? That is the key word for bar graph. What I've found in my years is that students tend to default to the bar graph, and it's not always the case. But anytime you're given more than one thing and you're comparing those things, that is going to be a bar graph. The line graph is used when you're tracking things. I drew an error bar here. We're going to come to those later. You can see my error bar is not very straight or good in any objective measure. But what would you use a line graph for? To track trends, right? Those two words, tracking, trends. Anytime you're going to think over time or over temperature or anything like that, when something's going to change, here we have sales over time, and it's a line graph. You're tracking a trend. Line graph, bar graph are going to be the most common graphs that you're going to see in this curriculum, and that's what they do. Over here we have a histogram. You won't see a histogram very often, but a histogram is used as frequency. It's counting, literally. You see this as a bar graph, right? So there's comparison here, but the y-axis is always going to be frequency. Every time it's going to be counting something. Right, and then scatter plot. Okay, we're going to stop here for a minute because scatter plots are really confused. First of all, it's not a line graph, so you don't have to connect the lines. A scatter plot is going to be used anytime you're making a relationship evaluation. What does that mean? So if you're determining whether or not there's a relationship between two seemingly unrelated variables. Is there a relationship between the age of a student and the number of tardies that they get in a given year? Two seemingly unrelated things, right? Well, you could plot the points on a scatter plot, and you could determine if there's a positive relationship, if it goes up, there's a negative relationship. When one goes up, the other goes down, or if there's no correlation. If there's seemingly no pattern there, you would use a scatter plot to determine that. Bar graph, line graph are going to be really common as far as what you're going to be asked to draw in class, but there are a couple of examples of FRQs where students are asked to draw a scatter plot. And so in the prompt, you're looking for those words of relationship. Researchers were attempting to determine if there was a relationship. As soon as you see those words, immediately think scatter plot. A couple other graphs that you may see, the pie chart. I love that this pie chart has no title or anything, so there's no way of knowing what the USA has 67% of and the rest of the countries have less of. But it is comparison to the whole. Comparison of one thing to the whole thing is what a pie chart is used for. The dual y-axis, this is where you have multiple things that are being measured based on a single independent variable. The independent variable in this particular graph is time, and you're measuring not only number of homes, but also the average price of those homes. Allows you to compare those two variables directly based on one single independent variable. A logarithmic graph, notice the y-axis here goes up in factors of 10. This is an exponential type curve, but the logarithmic graph allows it to be viewed in a more linear way. All right. No real anything here as far as like details for our purposes. For most purposes, it's just looking at the x and y and comparing those numbers. Just allows you to see it in a better way. And then the last one is a box and whisker plot. Box and whisker plot has to do with the comparison to the center, the median, and you have the four quartiles that come, two off either side. You have the medium or the middle 50%, and then you have the outliers, which represent the whiskers, and the box represented by the middle 50. I'm going to be honest with you. I have not seen a box and whisker plot on the exam. I have not seen a question based on them. It could happen. So I think as long as you read the prompt and the context, it'll probably come out pretty clear. Those are the types of graphs. Well, how are graphs constructed? Well, all of the bolded keywords or action words that you'll see in AP Bio are worth 1 point, except for the word construct, which is worth 3 points. Those 3 points are correctly labeled axes. Those axes not only need to be named correctly, but they also need to be, the unit needs to be listed next to them. You can see here the antibiotics are listed at grams per meter cubed or gram meters negative cubed. And I put the slash in there being silly. The yield is absorbance at 550 nanometers. Talking about light, I guess. So lots of weird stuff here, but you do have to have those units on there. The second point is going to be, are your plots correctly are the points correctly plotted? And how do you do that? Well, big thing that students will mess up here is College Board is going to give you like a square, right? It's going to have some like boxes in it, and you have to use that. Now, you can't use more than that, but you also shouldn't use a very small portion of that. You should appropriately scale your graph so that the data can be read correctly, and then use that scale in order to correctly plot your points. And then the third point is going to be, are your error bars correctly plotted? Notice this graph has an error bar on it where you have a particular point. Maybe this is 0.96 plus or minus 0.1. I don't know. I'm just saying, right? And so there's that. More about error bars in a moment, but those three things need to be plotted correctly. Hopefully your teacher will give you lots of practice on graphing so that you will be adequately prepared for that particular question on the exam. Every year, number 2 question has a graph on it, and that is worth 3 points. So it's pretty important. Another thing that you're going to be asked to do often is describe data, right? And it could be really simple. Sometimes the descriptions are really simple, like describe the difference between 2008 and 2010 when it comes to number of bears. There were 9 more bears than there were in 2008 than there were in 2010. That is a description of the data. It's very simple, right? But it could be make a claim concerning bear population and salmon population based on the data. And then you would say, when salmon escapement decreased, bear populations decreased as well. That'd be making a claim based on the data. You're describing this data. You're making heads or tails of it, right? You could say you're making an evaluation of it as well. That's another way you could think of it. Moving on to the statistical calculations. Now, real quick, first of all, you will be given an formula sheet, at least in my classroom. Can't speak for your teacher, whoever that might be, but you are given a wide range of formulas in order to use to do these sorts of statistical calculations. However, you're not going to need any of them. Let's talk about that a little bit. So go back to where we were. I have no idea how that happened. So the average, you've been calculating average since you were young, which is the same as mean. This equation is weird. and I don't understand it, nor do I need to. I know that I need to add the things together and divide it by the total number of things, and that's my average. You're going to use averages a lot in here. Median is literally the middle value. Count to the middle and find that's the median. And one of the things that I use these two values quite a bit. The median and the mean tell a lot about the statistics that you're looking at. And if the median and the mean are really far off, that means that there's probably a lot of variance in your data. A lot of variance in your data. And how do you measure variance? Well, variance can be measured by something called standard deviation. Standard deviation. You're not going to be asked to do a standard deviation calculation in this class. The easiest way to describe it is take the average data point, compare it to the average. How much does it deviate from the average? How much does one data point deviate from the average overall, right? Or you could just think of the amount of variance in a given set of data. Why is it important? I'll use one of my honors classes last year as an example. I had an honors class one time that had an 80% average on a test and an 80% median. And you're like, wow, it's really close. The standard deviation for that set of data was 17 or something like that, 17, 18, something like that. There was a lot of variance in that data. Even though the median and the mean were similar, they didn't really give you a good picture of the data, right? And so standard deviation is really important. Understanding it at the level you would have to in, say, like an AP Stats class or something like that, not important, right? So don't spend a lot of time here, but just a general understanding. Now, where you're going to see standard deviation more likely in AP Bio is this idea of error, standard error. Here's the equation. You're going to be given that equation. At the same time, you'd never have to solve for it. I don't know why we're provided with the equation, frankly. Whatever. There it is. You're never going to have to use it. But what you will have to use are these standard error bars. All right, so what does standard error do? So in this particular graph, we have jumping jacks and we have heartbeat. So apparently this is a scatter plot, right? Because we have two seemingly unrelated things, and we're determining a relationship. There's a positive relationship between jumping jacks and heart rate, because as jumping jacks goes up, so does heart rate. Easy. What does this little bar mean? Well, let's look at the 5 bar here. This 5 bar is at 89, so that may be my class's data. And it's 89 plus or minus 9, which goes all the way up to 98 and all the way down to 80. What does that plus or minus represent? The best way to think about it is if you took 1,000 classes, right, and you did this same sort of test, then you might expect that at 5 jumping jacks, there would be a range of 98 to 80 beats per minute. In another class, it might be 82. In another class, it might be 96. In my class, it was 89. This standard error gives you that range, right? Now, how are we going to use that number? Every single time that you're asked to draw a graph, the very next part is going to ask you a question, something like this. It's going to say, determine the difference, or something along those lines, determine if there's a difference between 5 jumping jacks and 10 jumping jacks when it comes to heart rate. How can you do that? Simple rule. If the error bars overlap, see how this error bar is at 98 and this one goes all the way down to 95. There's an overlap of 3. So what that's telling us is that somewhere, some class, somewhere, it is possible that 5 and 10 were the same. And so is there a statistical difference between 5 and 10 jumping jacks when it comes to heartbeat? No. You would just simply answer that question by saying there is no difference. That's it. What about 15 and 5? Well, 15 and 5 do not overlap, so there is a statistical difference between 15 and 5. There's no chance that they would ever be the same, therefore they are different from one another. That is how you're going to use that data in this class, and that is the extent of how you're going to use the data in this class. And so don't spend a whole lot of time here. Don't get in the weeds with standard error. You can do the calculations if you want. I don't know why you would. It's pretty intense, and it is not the goal of this or the scope of this class. Speaking of that, hypothesis evaluation is something that we will do quite a bit of. So a hypothesis, very common lab that we do in AP Biology that's all over the country, I'm sure, is this Oreo lab, where you're going to compare Oreos to double stuff Oreos. And the assumption is that double stuff are actually double stuff, right? That there's actually double the amount. That the assumption is that it is going to be what it says it is, right? And so let's say that we get some data. And well, first of all, that assumption that we're making is our null hypothesis. The null hypothesis assumes that there is nothing going on with the data. It is exactly what it says it is. Another example, the null hypothesis, if you flip a coin 100 times, is it's going to land on heads 50% of the time and tails 50% of the time. It assumes that there's nothing going on with a penny, right? What if there is? What if it's 30-70, right? Then you have to reject the null hypothesis, right? That this is not providing me 50-50. What if it's only 1.5 times, not double? Then it shouldn't be called double stuff, right? You have to reject that null hypothesis, and you have to come up with an alternative hypothesis. Somebody in the factory is tampering with the double stuff button. Somebody has weighted the coin to always land on heads. Whatever. Those are examples of alternative hypothesis. You have to come up with an alternative when the null needs to be rejected, right? If the data matches, and we're going to do a test called chi-square to determine whether or not the data matches, then you fail to reject the null. You keep the null where it is. Moving on. There's this bit here of interpreting biological meaning. This is just another way of assigning value to data. Again, you might be asked to do this as like a predict type question. Here's I like baseball, home runs per year from 1880 to 2020. What happens to home runs? Well, they've gone up significantly. My guess is that training has gotten better, food's gotten better, maybe steroids. You get the idea, right? I'm assigning value to this. I'm giving it meaning. I'm giving that data meaning. Could I go and then test those things? Sure. And so that kind of lends itself to more scientific method. But you'll be asked to do that. You'll be asked to evaluate things, sometimes giving it meaning, right? And when I say biological meaning, it's obviously going to be part of the concepts of the class. It's not going to have anything to do with home runs per year. It'll be some sort of conceptual work that you'll do with that. Okay, moving on. Argumentation. Mighty Mouse is there because this was a really small picture. And so you're oftentimes asked to make a claim, state the answer to the guiding question, make a claim concerning XYZ, right? You're going to provide evidence for your claim, or it's going to be like justify is where you're going to give justification. You're defending your choice of evidence, right? And so these are things that you're going to be asked to do quite a bit in here. And, using those action words, make a claim is an action word, justify that claim is an action word. And so we call this argumentation in general, but I believe that steroids were an important part of the increase of home runs from 1980 to 2010, justify that claim, and then I would give evidence for that, provide evidence for that, right? So very important, something you've been doing since you were young. So here's a couple of things just to look at, just some examples of things. This is actually taken from the 2025 FRQ. I don't have any questions associated with it, but I think I like this example because this stuff is not common sense, right? siRNA, section 2062 SI, it's not normal things, but it doesn't really matter, right? It doesn't really matter because you can look at these error bars and you can see that there's not a significant difference between SR protein, section 62 siRNA, and section 62 protein and siRNA, because the error bars, overlap. That's the kind of thing that I want to use this example, because a lot of times I think what happens in this class is students will get derailed by these words, and they will leave the concept behind, right? You'll leave this thing that's a bedrock and something that you learned and you can make heads or tails of really easily, and you will leave it behind for the sake of this data or this information that no one's going to teach you. We'll learn what siRNA is, but I don't know what section 62. There's a prompt that talks about it, but oftentimes the prompt is giving you way too much information as well. And so you kind of have to cut through a lot of the information in order to get to the real thing. So this is a perfect example. This is right off the FRQ from last year. Here's another example. This is the data table from last year, and just to kind of show you how would you graph something. This actually is helpful. The average general and oriented activity in male moths injected with saline or siRNA. So what are we doing with siRNA and saline? We're comparing the two, right? We're comparing those two things, and each one has a general and an oriented activity. And so saline, and what's our basis for comparison, right? What is our thing that we're manipulating? We're manipulating the saline and the siRNA. And what are we measuring? We're measuring their general and oriented activity. And so this is going to be a bar graph, right? And saline is going to be on the bottom, or the x-axis, and siRNA is going to be on the x-axis, and each one is going to have two bars that are going to correctly represent that, right? And you have 95 plus or minus 5, you have 98 plus or minus 8, and so this data is going to go, you're going to have data that's going to go from 25, 20 to 100, right? So it's going to be pretty wide range of data there. And so just an example. This is right off the FRQ from last year, but this is the sort of thing that you're going to see and the kind of thing that you need to be able to make a graph of. Practice question. Here we go. Researcher measures the growth of two bacteria cultures, A and B, over a 12-hour period. The mean population size of each time point are shown below with error bars representing the standard error of mean. Based on the data, which of the following is most reasonable conclusion? So let's look at the graph without looking at ABCD real quick. We can see that culture A and culture B are growing fairly similarly up until around 6 hours or so. How do we know that? Well, here the error bars seem to be overlapping, and here they don't anymore, and then they go apart. Let's find that in our prompt. The difference in growth rate between cultures A and B after 6 hours is likely statistically significant. A is the correct answer. I'm not even going to read BCD because they can't be right if A is correct. It's a good test-taking skill. If you read BCD, you're just going to make it more muddy. Keep it clear. Choose the right answer. A is the correct answer. Hopefully this was helpful to you. If it was, watch 0.2 because, well, it's probably going to be released soon after this one. See you later.